Vinegar is a widely used product both in food preparation and for cleaning. It is made of acetic acid and water, which provide it with an acidic pH, and because microorganisms cannot survive these pH conditions, it can be used as a cleaning agent. Here we're going to explore the process of vinegar production, its relevance in the industry, and its relationship with microbiology.
The process consists of three main parts: saccharification, alcoholic fermentation, and acetic acid fermentation. Each one is essential for the production of vinegar. In the saccharification process, the starch, which is a polysaccharide, is broken down into small sugar units that can be fermented. This process is carried out with the help of some enzymes of microorganisms like Aspergillus oryzae. After saccharification, the sugar units are fermented to alcohol. In here, microorganisms play an important role. Saccharomyces cerevisiae is a yeast able to carry out spontaneous fermentation that turns sugars into alcohol.
After fermenting the sugars, the alcohol produced can be converted into acetic acid with a second fermentation, in which alcohol is oxidized to obtain acetic acid. This process involves two main microorganisms, Acidobacter species and Gluconacidobacter saccharivorians, with an optimum pH between 5.6 and 6.3. The last step can be divided into two separate substages, where the different microorganisms interact with the alcohol. In the initial to mid stages, the Acidobacter species do the most fermentation, but as the acetic acid concentration increases, these microorganisms die and leave the Gluconacidobacter saccharivorians to perform their remaining fermentation. This microorganism can survive as it is more resistant to acetic acid. However, as alcohol becomes more limiting, it starts dying. Vinegar shows very particular characteristics. As this product has reached its maximum state of oxidation, there are no detrimental organisms that can make it go bad, making its shelf life indefinite. A big worry for many food producers is the pathogens that may infect the food and potentially infect the consumer. However, vinegar is very particular and it has actually shown effectiveness against infectious pathogens such as E. coli and Salmonella.
It has shown such an effectiveness that many people use this product for cleaning purposes. The production of vinegar has evolved throughout the years, and it's important to outline its relevance in the food production industry.